So this is the second part of the complementation test that we're discussing about, and in this video we'll talk about the mutants of T4-phage. T4-phage, as you all know, it's a bacteriophage and it is a lytic phage. It's virulent, which kills the cells, bacterial cells, when it infects. Now, in T4-phage, a series of mutants have been identified which are called as amber mutants. But these amber mutants are so called because they introduce a stop codon. So, as a result, the transcript terminates prematurely and you don't get a functional product. Now, these T4 phages have been very useful in gene mapping. Now, here in this board, what you are seeing over here is I'm talking about two mutants, amber, they are called as amber B17 mutant and amber 18 mutant. Now in amber B17 mutant, we are discussing two genes, gene 18 and gene 23, and in amber E18, we are discussing about gene 18 and 23 again. The difference is in B17 mutant, the mutation is in gene 23, whereas in E18, the mutation is in gene 18. Okay. Now, this DNA makes RNA, this makes protein. When gene 18, which is normal, it makes a normal product, and gene 23, it has a mutation, so it makes an incomplete or a truncated product over here. On the, now, this because gene 18 is normal, so tail fibers are formed normally. Here, what happens? Gene 18 is mutated, so the product is mutated over here. Okay, it's truncated. Now gene 23 is normal, so it makes normal transcript, normal product. Gene 23 is responsible for formation of the head. But because in the same protein, now these two mutants are actually transfected together. Because they're transfected together, so the same protoplasm, you will have the tail fibers as well as the head. Okay? Now this head, and so they form a normal phage. Okay, so they form a normal phage which causes lysis because of complementation. Now, one thing which you have to understand over here is this experiment is done in E. coli B cell. B cell is a restrictive host. The meaning of restricted host is that it will only allow the wild type phages to grow. The mutant phages cannot multiply, they cannot grow. Now let us consider a second situation in which we have taken the same situation except that in both these mutants, the mutation is in gene 23 over here. Here also it is in gene 23 and here also it is in gene 23. Now as a result, this is making a normal tail fiber and this is also making tail fiber. The head proteins are mutated. Now this will not cause any lysis because there is lack of complementation. Now, this is now what you should understand over here is that complementation is different from recombination. Complementation doesn't tell us about the genes which are linked, whereas recombination will tell us about genes that are linked. Okay, it's not that recombination doesn't take place in phages. Recombinations take place in phages, and we have an experiment to show this. Now, this experiment is again the amber mutants. Here you can see that there is a gene which is amber B17 and this is amber B18. Now there's a mutation over here and there's a mutation over here. Now both these are transfected in the E. coli cell. This strain is CR63. This CR63 cell, it's a permissive host, so it will allow all kinds of phages to grow, whether they're mutants or they are not mutants. So this replicates, okay, so this is replication over here. So this replicates, have many progenies, this replicates, you have many progenies, right? Now, in the same protoplasm, it is also possible that you have a recombination, breakage and joining. So you have a recombination that's taken place over here. Now with this recombination, here you can see that this is the wild type strand where both the mutations are absent, and this is a double mutant. You can you can count the number of mutants and double mutants in phages by, because this is a permissive host. To count the percentage of wild type phages, what one has to do is one has to grow them again in a restrictive host. So when you grow in restrictive host, you can count the number of wild type phages that are present. Now, how do you calculate this? Is 40, 10, 10, 10, 40. How do you calculate this? If you get the number of wild type phages, you know that equal number of phages will be double mutant. Okay, and when this is double mutant, then you can find out the other percentage. You can add this and minus subtract by 100 and divide by 2, so you know individual mutant progenies that are present. So this is how one can do analysis, or one can identify, one can know about the distance of the genes. Now complementations are done in one second. So complementations are done for you, or you can do complementation analysis for most of the genes. However, genes which are there are double dominant mutations, you cannot do. Okay? If there are point mutations, again you have some problem, because there is a phenomena that's called as intragenic complementation, where if there's an amino acid substitution. So if you have so if you have two polypeptides forming a functional product, okay. Now if there is a mutation in one of the polypeptides, so let's say there's a mutation, okay, then you have they don't form a proper functional product because of the mutation, so you don't see any function. Or it can also be the other way around. There's a mutation over here, so if they don't allow you to form a functional dimer, so they don't function. However, it is also possible intragenic is if this a mutation like this. If you have a mutation this way, then they give rise to a wild type product. Okay, so this wild type product, it can be completely functional, it can be a hypomorph, that does not matter. But then this kind of a situation also takes place. So therefore, there are some limitations of doing complementation test, where you cannot use them for dominant alleles. You cannot. It's very difficult to conclude results when you have point mutations, but they're very good for frame shifts, deletions, and so on. So this is about complementation test. In the next video, we'll talk about the R2 locus, which was a landmark in many ways that we'll discuss uh, in the journey of science. Dr. Seymour Benzer, he is a name which is very famous, and he was the one who used these kinds of recombination mutation experiments to identify many different loci of the R2 locus. Thank you.